So let me introduce the next speaker, Joachim Frank. And as I mentioned at the very beginning of my earlier talk, that the background of your family is really crucially important. So actually his father was a judge, and also his mother came from a distinguished Microscopy family who has social ties, including a friendship with Goethe's families, and he had 3 siblings. And almost the very same level, like how it was with Danny's family, the natural curiosity of his career was extremely important. And actually, he collected bottles filled with various chemicals, including hydrochloric acid and gasoline, and even explored the process of producing gas by heating coal. So his experiments resulted in a big explosion. And luckily he survived, but there is one secret he mentioned at that time, that the only secret when you do experiments at home, kept secret from your parents, because otherwise you're not sure you can continue your research. But luckily, after this kind of experiments, he went to a school, and he learned really a lot. And in a high school, he developed strong interest in science classes, especially physics. And of course, there were more experiments. He always wanted to find really new things. So he introduced building crystal radios, which actually was as miniature as radios that fit into soap boxes. And after that, he continuously did his kind of research. So he had the university in Germany, at the University of Freiburg, after he moved to Munich. And later on, for his master's degree, he worked at the institute to develop actually new techniques by electrons on liquid gold. After that, he received a very prestigious fellowship from the Harkness Fellowship, and he moved to the US. And after his research there, he used the most advanced image processing technology in the world at that time. So his academic journey was also at Berkeley and Cornell as well, and after he returned to Germany. And he was the very first who had in the 80s a first 3-dimensional cryo-EM reconstruction of an E. coli ribosomes, which actually changed the world understanding of this kind of 3D structured. And as I mentioned with Danny Shechtman as well, that if you find out something new, there is always a high chance not to recognize at the very beginning. And actually he submitted his paper to the top journals first, to Nature, Science, and other one, and he were continuously rejected from all the journals, but he didn't stop. And finally he published his work actually in an ultramicroscopy journal, which at the time it was not a top journal. However, he decided to publish it, and later it was clear to everybody that this kind of multivariant analysis of the images can change our understanding, not only what he looked at that time, but almost all kind of chemical structures. He also worked in Albany and the Wadsworth centers, and also developed the spider image processing system. So it's not surprising that because after his discovery, the atomic structures could have been visible by the electrons, that the Nobel Committee awarded him a Nobel Prize in 2017 in chemistry for discovering the cryo-electron microscopy techniques. And of course, not only this one where he discovered, he included many other discoveries in his life, the first 3D visualization of ribosome bone transfer RNAs, and I could tell, of course, after that, how many new things he delivered to the world. His current position, he is currently at Columbia University, and he is in a department where 3 Nobel laureates are there. So I wish maybe once at Semmelweis University we have such a department where he can work. Of course, he received plenty of awards and honors, but again, when I asked him, you know, what he is the most proud of, he actually showed that, of course, the family. He has 2 children, and all of them are really has a special knowledge. But I am also asked by plenty of people that, okay, if I became to be scientist, I need to forget everything what I did in my career because I have to concentrate only on science. But it's not true. So if you look at who is Joachim Frank, you can also find brilliant articles, fiction articles. So not only in the scientific field, he did really, really huge amount of work and things, but also in fictions. He's an author of several fiction books, which is actually now translated also to many languages. Concerning photographies, actually he's a brilliant photographer as well, so he has really, really, really many hobbies. And also when I go further, it's also very visible that he not only likes the books, but also classical musics. And also I learned from a couple of days that he's more of a white wine person than a red wine one. Of course, he also has a wisdom for the young, and he always says that keep your peripheral vision open. Don't just follow a specific plan because you just don't know what opportunities come along. And if you keep your eyes open, you always find something interesting. So Joachim, it's a great privilege for Semmelweis University as us to welcome you. Thank you for coming and spending your very valuable time with us. Thank you very much, Joachim.
Thank you very much for this wonderful invitation and laudation. So I wanted to talk about the technology that I have helped develop over the past decades. Cryo-electron microscopy, a new foundation for molecular medicine and drug design. So today's medicine is guided by the knowledge of the way biomolecules are built and the way they act in concert in the cell. With that knowledge, effective drugs can be developed, small molecules that are designed to bind to target molecules and thereby change its behavior. And this, in a nutshell, is the definition of molecular medicine. Knowledge of structure, dynamics, and organization is required on different scales. And these are associated with the fields of structural biology and cell biology. So I'm most fascinated with molecular machines in the cell. In 1998, Bruce Albert published a very visionary article in which he stated essentially that we have to rethink cell biology. It's not just chance encounters with molecules bouncing into each other, but rather these are processive actions. These are processes that involve molecular machine. So there's a beginning, and a middle, and an end. And these are processive processes where a ligand bind stays on the molecule and then leaves, and we have a certain direction of time. And so many molecules act in concert in a processive way. We wish to know the structures of all components, but also the way they interact dynamically. And we need a targeted drug development to combat diseases. This is where reductionism comes in. You saw a picture before where all the different molecules sort of are compressed in the cell, and it's very difficult to look at them in detail, and that's why where reductionism comes in. You have to extract them from the context of the cell to study them in detail. And we need atomic resolution. It's very difficult to get atomic resolution while the molecules are still in the context of the cell. And structural biology, it's a concept that formed in the last decades it's a field that mainly focuses on the structure of biomolecules in isolation. So what are the tools of structural biology? One is X-ray crystallography. Molecules can be made to form crystals. They arrange closely packed in regular order. And in that case, the atomic structure can be determined by X-ray crystallography. And here we have Max Perutz and his student John Kendrew. In 1962, they received the Nobel Prize in Chemistry. They were among the first protein crystallographers. And they studied myoglobin and hemoglobin. And here they're shown with a model of hemoglobin. So the other tool is electron microscopy. Well, why do we need electron microscopy? Why do we need another technique? Well, obviously, many molecules don't form crystals. Even if they form crystals, they don't form highly ordered crystals. So we are unable to get to atomic resolution. So this is where electron microscopy comes in. And the particular electron microscope that I'm talking about is a transmissional electron microscope, which means that the electrons go through the object. So the object is transparent. Object has to be very thin. And transmission means that a projection is formed. So we don't get a three-dimensional image, we get a two-dimensional image. And the closest parallel is probably the jellyfish. That's an everyday object where we see all the way through. And so what we see is determined by how much material the light goes through. And the electron microscope is an optical instrument, but unlike a light microscope, it has magnetic lenses and operates in a high vacuum. Electrons are produced at the top and then accelerated to a very high speed, and then they travel down the column. And what you see first is the condenser aperture, condenser lens. That's the illumination system, produces a focused beam, and then the sample is immersed in the objective lens. Objective lens is the most important lens that determines the quality of the whole imaging process. And then an intermediate image is formed somewhere between objective lens and projector lenses, and that's further amplified and enlarged by the projector lenses. And in the end, we have the system of detectors. Nowadays, these are electronic detectors, electronic cameras. In the old times, one actually sat at the electron microscope and looked through a binocular on a fluorescent screen, and pictures were taken on plate camera photographically. So three-dimensional reconstruction is necessary because we have only two-dimensional projection images and which are formed, and we can form a three-dimensional image if we take a series of two-dimensional images in different directions. And electron tomography is a particular technique where the object is tilted into many increments so that a full range of two-dimensional projection is formed. And the pioneering work in 3D reconstruction was by Aaron Klug and David Durossier, and they made first computerized reconstruction of a bacteriophage tail using a particular approach, a particular software approach in 1968. So there is a particular Fourier theorem that tells you how to collect the information and how to put it back together. And so the single particle techniques that I came up with, they asked the question, you know, why do we have to tilt molecules? And in the process of tilting the molecules, we're burning the molecules more and more. We collect radiation damage, and in the end, the molecule is maybe we got a 3D reconstruction, but it is absolutely meaningless because meanwhile the molecule has burned to cinder. Single particle techniques take a radically different approach. Why not make use of the fact that that molecules are randomly dispersed, they're randomly oriented, so I don't have to tilt them. They have already tilted themselves. Okay? There's no tilt series is necessary. We just take snapshots and make use of the fact that we have many, many angles already available. And so the trick is simply to find out what these orientations are. So the structural information comes from images of single, which means unattached molecules that exist in many copies. And they're randomly oriented. Molecules are free to assume all naturally occurring conformations. And a single snapshot may already give us hundreds of particle views. As we collect more snapshots, more orientations will be covered until we have enough to reconstruct the molecule in 3 dimensions. So that was the idea. At the time, it was completely radical and nobody believed it was going to work. And today, it's the only way of getting molecular structure. The concept may sound very nice, but it really postpones the problems to after the fact. After the experiment is done, we need to solve many problems, and they all have to be solved with software, with mathematical technique. So we have to line molecules, we have to find the angles after the fact, we have to sort them into different classes depending on the way they look. So all these techniques had to be put together. So I designed a workbench of programs. So there are something like 400 different programs that all had to work together, and one was able to build a workflow from different elements. So the first success of this was in 1987. We were able to do a reconstruction of the large subunit of the bacterial ribosome. And here it is presented in a big wooden box as a series of slices that could be put onto a light box. So you could see it from different directions. And at the time, this was not a cryo techniques. This was a molecule that was negatively stained, because at the time there was no cryo methods available. So the cryo-EM was really developed through different pathways. The single particle techniques I developed, and then Dubochet came up with the cryo methods, and then these two techniques merged at the end of the 80s, and that's when cryo-EM was formed. So here's the cryo part of the cryo-EM, which works by plunge freezing of the molecules. The molecules are in a buffer, and then a little droplet is put on the grid. And then comes a blotting, which reduces the layer, the liquid layer, to very thin, so that electrons can penetrate. And then there's a step of very fast plunging, freeze plunging, into liquid ethane, which is on temperature of liquid nitrogen. And if the speed of the plunging is fast enough, then a very new form of ice forms which is called vitreousized. It is glass-like, and the water doesn't have the chance to form crystals. And this unique form of ice is just right for preserving the molecule in its initial native state. And that was really the breakthrough. And here's sort of a more realistic presentation of this, but you have to imagine that this is now a computerized contraption which costs by itself like $50,000 or so. It's completely automated. And this is the result of such a freezing process and imaging afterwards. You see a salt and pepper kind of appearance because we have to be careful with the electron dose. You have to keep the dose very much down in order to avoid damaging the crystals. Damaging the molecules, I'm sorry. So these here are the first images that were at the time of the confluence of the two techniques. Single particle technique, which had nothing to do with cryo, and the cryo technique of the Boucher. And this was here sort of the golden time, you know, for the first time we saw all these molecules for the that had never been imaged by X-ray crystallography because they could not be crystallized. But at the same time, the resolution was very, very low. So we could only hope to show molecules in their naturally occurring conformations and make sense of them by using existing X-ray structures and mold them into it. So at that time, we didn't have any hope to get to atomic resolution. And that really took years from there. You see the years 1995, 94, long time ago. So these are now the incremental steps of what was called later resolution revolution. First of all, the whole concept of single particle cryo-EM, which in itself didn't look like it would ever get to that place. Then came electron microscopes that were interfaced with a computer. That was a very big step because it enabled data collection, automated data collection. And then came much faster, more powerful computers. And then came a technique called three-dimensional classification, the extraction of multiple structures from a mixture in a sample. So you could no longer, you didn't have to purify molecules to a single species anymore. You could just have them partially purified, and you could, with these techniques here, with sophisticated software, you could pull out all these structures at the same time. Okay, so I call it story and a sample. And then there was a huge improvement of image recording quality by the switch from film to electronic camera in 2012. The direct electron detection cameras. They were introduced by 3 different commercial companies. So all of a sudden, all these techniques that already existed and all the mathematical apparatus was there, finally came to fruition because a very simple obstacle was removed. The obstacle was a very poor quality of recording, which was recording on film. All of a sudden, recording quality went up the roof and enabled us to get to resolutions of 3 angstroms and then 2.2, and the best resolutions are even 1 angstroms now. These are the big machines that exist now. Here's a Thermo Fisher one, and here's a JOL one, and they're now spread out all over the world. And here you see the kind of recording quality. You can see that the image is a very high contrast. And we now have this kind of workflow from protein purification. Then we still do negative staining in order to see is there anything there, because electron microscopy time is very precious. So we first look at some kind of a screening, and then we do a go-ahead and do the actual cryo-EM recording. Then we get thousands of images, and then go along with the process of 3D classification. And then we get a three-dimensional electron density map, which is a three-dimensional image. You have a density in every single point. And that image then is the basis for atomic modeling. So you can, looking at the signatures of amino acids, we can identify, we can go along the protein chain and can make the exact determination of the different residues. And here I show you the image of the ribosome in 2016, where we, for the first time, took advantage of this new technology. You see the crisp details, you see individual helices. The false coloring is used to show you where the highest resolution is. It's in the interior. Outside, there's more flexibility, but in the interior, there's a resolution of 2.3 angstroms. So nowadays, if you type in cryo-EM structures into Google, you get pages and pages of structures, you know, because meanwhile, this is the way of getting atomic structures and no longer X-ray crystallography. Many X-ray crystallographers have switched to cryo-EM because it is more effective. It gives you more lifelike structures, so they're not sort of in a straitjacket, but rather they are lifelike in the native environment. So with the knowledge gained by high-resolution cryo-EM, the origin of many diseases can now be understood, and effective cures can be developed. Vaccines, antibodies, antitoxins, antibiotics, all can be designed based on the knowledge gained by cryo-EM. And small molecule drugs can be designed that stabilize the target molecule and enhance its function, suppress its function, for instance, by changing its dynamics, or blocking its interaction with a functional partner, or change or modulate its function. And the development of cures for infectious or insect-borne diseases like malaria, tuberculosis require the detailed knowledge supplied by structural biology. And I'm giving you 4 examples of cryo-EM advancing molecular medicine. One is the human CDK activating kinase, CAK, which offers the potential of drugs in fighting cancer. Cystic fibrosis, transmembrane conductant regulator, CFTR, the malfunctions of the channel caused by mutations and offers target for effective drugs. Ryanodine receptors, RYR2, which are implicated in heart diseases and show many mutations, and the identification of the site of these mutations is a major help in the drug development. And then COVID-19, the spike protein of SARS-CoV-2 virus, the structure dynamics of interaction with human host and antibody mapping. So cancer, here's an example where the knowledge of this particular structure has given clues to combating cancer. So it acts as a master regulator of cell growth and division. It regulates transcription initiation and the cell cycle by phosphorylating RNA polymerase II and cyclin-dependent kinase. And due to this central role in cellular physiology, the CAK has been identified as a promising target for cancer therapeutics, and it's a possible target for antivirals. Here's a structure in resolution range of 1.8 to 2.3 angstroms, which is really a resolution range in which drugs can be effectively designed. Cystic fibrosis. Here's the molecular structure of the human CFTR ion channel. And we can now identify the positions of the cystic fibrosis causing missense mutations. You see how many mutations are there spread out over this entire molecule. And just a few weeks after I received my call from the Nobel Foundation, I got this message which touched me very deeply. Dr. Frank, I want to add to the chorus of congratulations that you have no doubt received since the Nobel was announced. I have a special reason to send a thank you as well. My 7-year-old grandson, Jackson, was diagnosed with CF as a newborn in 2010. I have attached news that we received this week regarding his 10 months on combination therapy. I just wanted to share this as a small expression of my and Jackson's gratitude. So this was a total surprise to me because I didn't make the connection. You know, I had not made the connection that I could personally be thanked for this development. But then thinking about it, yes, it has contributed to it. So I was very deeply touched by this. Ryanodine receptor RY1, skeletal muscle, and RY2 for the heart. So we've been working on RY1, which is very closely related to RY2 initially. So we're among the groups that got the first atomic structure in 2016. And I collaborated with Andy Marks and Wayne Hendrickson at the time. They, as groups, tried for 7 years to get highly diffracting crystals, and we're unsuccessful. They only got to something like 9 angstroms resolution. And after we attempted to use cryo-EM, we got to the point of 3.5 angstroms within a few months. And here again, I show you positions of mutations that are associated with malignant hypothermia and various myopathies, all in ryanodine receptor RY1, the skeletal one. And here are pathogenic mutations of the heart muscle ryanodine receptor. You see the numerous locations of mutations that can now be effectively addressed with the knowledge of the structure. And the diseases implicating mutations of the ryanodine receptor of the heart are a central core disease, a central nuclear myopathy, multi-mini core disease, congenital fiber type disproportion, and so forth. And then finally, I'd like to tell you about COVID-19. And this is a very unusual story because just 2 weeks after receiving the genome sequence of the virus from Chinese researchers, the team had designed and produced samples of the stabilized spike protein. And it took about 12 more days to reconstruct the 3D atomic structure of the spike protein and submit it the manuscript is Science, which expedited its peer review process. So in March of that year, we already had the structure, and that was essentially gave a guideline for the development of vaccines and looking for the place where antibodies effectively bound. So there was the beginning of a whole era where cryo-EM was absolutely instrumental in fighting the disease. Here's one of the articles that was published at Columbia on potent neutralizing antibodies against multiple epitopes. They were isolated from patients that had survived a serious illness. So coming toward the end, the EMDB, which is a data bank of three-dimensional cryo-EM reconstructions, has really risen very, very fast. And you can see this is the annual depositions, which has become very flat for X-ray crystallography, but it rises exponentially now for cryo-EM structures. And in conclusion, in recent decades, medicine has become very powerful by leveraging the knowledge of molecular structure. We speak of molecular medicine. And X-ray crystallography has been the traditional technique to determine the structure of biomolecules, but many molecules do not form crystals, so the cure to many diseases was completely out of reach. And cryo-EM, which does not require crystals, is a new transformative technology for imaging biomolecules and determine their structure in their native states. And many biomolecules, such as ion channels and receptors that are hard to crystallize for X-ray crystallographic studies, can now be imaged at high resolution. And based on the knowledge of the structure of biomolecules, drugs can now be developed to combat many diseases for which there was no cure. And here I show my team in 2018. My group is much smaller now. I have sort of a small leftover group that specifically uses a new technology, which is time-resolved cryo-EM, where we can follow reaction in real time. And I just wanted to show you the website. When you go to my website, you can find a lot of very useful things, including all the references that have any relevance here. But I encircled the one feature which is called Nobel 360, which is a place when you go to the link, then you get 360-degree views that you can steer, and you can see the microscope and the different places in my lab, and even a place outside of the lab, close to George Washington Bridge. And as Peter told you, there's a lot more than science that I've been doing, and you are welcome to look at this website, and has many stories, photography, and all kinds of musings. Thank you.
Thank you, Professor Frank, for this really inspiring and thought-provoking lecture. We have no time for one question. Any questions there? Students, lecturers? All right, then, Professor, since there are so many students here, what would you say is the one or two best advices for a young career researcher to maybe follow in footsteps like yours or similar?
Yeah, for me, it's always the idea of peripheral vision. Go through the world with open eyes and think about anything that you encounter as possible answers to your questions. Okay, the questions are, of course, when you're looking at a certain problem long enough, then it becomes integrated somewhere in your brain. It becomes a separate process, and that process is going on no matter what you do. And then you go through your day, talk to people, see something, see a flower and whatever. And the trick is that you're trying to see whether these have answers to your problem. You know, the answers can be very far-fetched, but I discovered as I went through my career that very often these peripheral perceptions had a lot to do with solving my problem.
Thank you very much. Professor Frank, it was really an absolute privilege and pleasure. To commemorate this event and also your contribution to society, I would like to present to you this Award of Excellence.